Pramod, and I am from Central University of Kerala, and I'm an associate professor in the Department of Plant Science. And first, I would like to thank the organizers, Peter and others, for giving me the opportunity to present here. And I will start with an introduction and then go right into my topic. Okay, so hunger and undernourishment can be considered as a serious problem, and you can see most of Africa and many parts of Asia and some part of South America and all these are under severe hunger issues, and also it goes in hand with undernourishment. You can see that here over the years the hunger index has reduced, but recently after 2020 the undernourishment issue has increased compared because of multiple issues including food prices, poverty, human conflict, and climate change. So one problem why we are facing this undernourishment and hunger is because of our less diversification of crops. We are using only very few staple crops like rice, wheat, maize, and we are majorly dependent on these crops. But there are many other crops which are underutilized and which are used in many communities. As you can see here, millets as a major source of nutrients, and it is heavily underutilized. And there are lots of root crops which are consumed across the world, and they are also having very high nutritious content. And there are several pulses which are underutilized and not even used that much. And lathyrus, if you take, which is the topic of today, I am going to talk about lathyrus. It's also an underutilized crop. It's coming under the list of underutilized crops, and it's considered as an orphan crop because very little research efforts are going on this crop. But it's a smart crop. I will tell you why it is so smart. Okay, it's commonly called as grass pea, and it's an annual cool season legume, and it's cultivated from ancient times for food, feed, and fodder. It is cultivated actually entire Europe and also Asia and also in Africa, and also in South America and some part of Australia. But if you see why it is smart, I can say here it's a climate smart crop because it has a high resilience to biotic and abiotic stresses, and it can come up very well in poor soils. Like minimal inputs are required, and it can give you a good produce even you don't put any fertilizer or anything. Another thing is it is a legume. That means it's a nitrogen fixer, which is a major part of sustainability of human population on Earth. Nitrogen is a major energy requiring material, which where plants, if they can fix nitrogen, they are very valued. And legumes, definitely, they add to our existence. And there are the morphological features suggest that it's a very penetrating root system, and it produces very high biomass. So narrow leaves and all adaptations which are required for adverse weather conditions. And nutrition, you can look at this crop and you can say it has a very high protein content, 28%, which is you can say next to soybean in terms of protein content among pulses. And it has a very high lysine content, threonine and homoarginase is a kind of a amino acid which is not present in many other crops. And also it is rich in minerals like iron and phosphorus and vitamins B1 and B2. And also it can be considered as a pharmaceutical or nutraceutical, like because L or homoarginine, which is very important for cardiovascular health, and it can be utilized as a medicine for all these cardiovascular health issues. And the last one which I'm going to talk is the beta ODAP, which is kind of a poison because this has been found as a poison because when people started consuming this crop for a long time during famine kind of situations, large quantities, people developed irreversible paralysis of lower limbs. And that is called neuroletharism. And beta ODAP is associated with neuroletharism. And basically, when you consume this pulse for a long time, that means 2 to 3 months, and for large quantities, that means you consume only this crop. Actually, in famine kind of situations, there is nothing else to eat. So you keep eating this pulse. At that time only you develop this kind of limb paralysis, which I can say. But this caught attention of people in early 1960s, and which where they worked on the biosynthetic pathway leading to the production of beta ODAP, which is beta oxalyl diaminopropionic acid. Actually, this pathway starts with the amino acids asparagine and acetylserine, and cysteine synthase is the first enzyme which converts it to an intermediate which starts this pathway, that is DAA, and DAA is converted to multiple steps through to DAPA or diaminopropionic acid, which is shown here, and which is finally converted to beta ODAP through two enzyme actions. One is OCS, which convert oxalate to oxalyl-CoA, and the oxalyl-CoA combined with DAPA with the catalysis of an enzyme called BOS or beta ODAP synthase to form beta ODAP. So this pathway is kind of known by biochemical analysis from 1960s and 70s, but only recently we know have the genome information about this crop. Actually, the first genome published genome came from our group, which is in 2023. In the same year, another paper came, which was published by John Innes Center, and they also published another draft genome on this crop. And recently, we have a chromosome scale assembly for this genome of Grass Pea, and which again came from John Innes Center. So now we have a very good information about genome aspects of this crop, which actually helps us to identify genes and clone genes, and also manipulate the pathway. And one particular thing I want to mention is Nature Communications paper, which came from John Innes Center. They identified, actually characterize the pathway, the final step in the pathway, that BOS. Actually, there is another paper which came 2 years later, which also talked about this particular enzyme, BOS, and they actually further characterized this enzyme. And they said that OCS and BOS form a kind of a metabolon, which lead to the production of diamino beta ODAP. So these are the final steps towards ODAP. So we can modify these enzymes by any of these genome editing methods or any of the other mutation techniques, and then we can actually bring low ODAP plants, which is more acceptable to the world. And there are so many RNA sequencing studies also, which helped us to get to the gene sequence and all those things. One of those such studies is done by our group. Here I am showing that we actually compared RNA, RNA profile of one low ODAP and a high ODAP cultivar, trying to understand how this ODAP affects the the metabolism in this plant. You can see Pusa, which is 0.2 to 0.3 ODAP, and whereas Rattan has 5 to 6 times less ODAP. And we looked at these plants, and we actually looked at its ODAP profiling, and then we went with the RNA sequence and then came up with a set of genes which are differentially expressed. You can see the ODAP profile. This Rattan and always has a reduced ODAP when this is seeds germinated and different time points of germination. You can see 5 days, 10 days when the seedling grow, you can see the ODAP levels are down, and then we can have, we profiled these particular stages and then came up with differentially expressed data sets. And we conducted MapMan analysis and found that genes related to asparagine, methionine, all the amino acids, and also carbohydrates, hormones, and stress genes, and are differentially expressed in these pathways, and which always associate, because low ODAP content rattan has a reduced stress gene expression, and which we try to correlate with APA levels actually. So we found that PUSA has if you apply stress on PUSA, you can get high ABA levels, but whereas rattan has not that much significant change in ADAP expression. And basically, what happens is when ABA content is low, and which affect reduced ODAP levels in rattan, and that will reduce a moderate stress tolerance in this group. So basically, beta ODAP helps in stress tolerance in this group. That is what our analysis says, and this paper was published in 2022. Actually, that is one of the aspects we worked on, and we are continuing working on that to understand the stress in this group. But another aspect which I wanted to share with you today is genome editing for function in this group, because we know many of the genes, and now it is the genome editing era, and we wanted to focus on these genes and try to see where we can manipulate this pathway and bring a low DAP variety, and for which we use the genome editing approach, where we targeted particular genes in the pathway using guide RNAs specifically designed, and use this which associated Cas9, and this will target these genes, and they will be edited. And basically, we looked at oxalate-CoA synthetase, which was the known gene at that point of time when we started the work. Beta ODAP synthase was not known at that point. But if we can reduce or convert, block the formation of oxalate-CoA from oxalate, that will reduce the ODAP content, because oxalate-CoA is a substrate for production of ODAP. That is also our idea. So we actually designed gRNAs and for this, and we usually we use agrobacterial tumefacience method also, but then legumes are generally recalcitrant, and we tried for a long time getting agrobacterial tumefacience, which was quite difficult for us, and so we have finally resorted to agrobacterium rhizogenous, which is hairy root method, and we developed a method for this plant, and actually we use the sgRNA, and you can see a single guide RNA approach we used first, where single guide RNA with a Cas9 in the vector we used, and we produced hairy roots, and we analyze, we actually use these flanking primers and amplify the region of the target site, and then sequence that, and then usually hairy roots produce chimeras. So that is one problem associated with hairy roots. The editing is never complete. You get a mixture of edits. So we have to, after Sanger sequences, we have to actually use some software analysis to identify the editing. And basically, whatever we tried, 15 root analysis, and you can see the edited gene sequencing here, and you can see most of the roots are edited, and the editing percentage is up to 73% editing, you can see in individual roots, and this is the range at which we can get these things. So we can see a very robust editing happening in these roots with this approach. But the problem with this method using single guide RNA is we have to go for sequencing to identify these edits, and to ensure that these hairy roots are edited, and which we can use for subsequent analysis. To overcome this problem, we went with another approach where we have used two guide RNAs. But if you use two guide RNAs, then we can actually, these two guide RNAs are selected 100 base pairs apart. So if you have that kind of an editing, when this editing can be confirmed by a PCR analysis. Here I am showing you that kind of an analysis where I have in the vector two guide RNAs. You can see two guide RNAs here, and then Cas9 is there, which is driven by U6 and U29 promoters, and Cas9 is by 35S promoter. And you can see the PCR analysis here. This first lane which I am showing you is the control line, control or vector control lines, where I have amplified the target region with the flanking primers. You can see around 600 base pair is amplified. Where in edited roots, you can see the smaller sized bands here. and here and again here. You can see a smaller size band, and there is a fade band here, which shows that certain percent editing is happening in these roots. And these, these bands are sequenced to see the deletions, which is large deletions which happen because we have used two sites which are on 100 base pairs apart. And basically, this editing or the deletion which we have seen is not is in the region of this target size, but not exactly where this target size. That is because when the homologous non-homologous recombination which repair these double standard break, they will do it in their own way, so we cannot actually predict what kind of decisions we can take. But what do I want to say is the double guide RNA approach actually can detect on the PCR analysis, and then we analyze for function these routes. And you can see when we have edited the oxalate content actually keep increasing. That means it blocks the conversion of oxalate to oxalate-CoA, and which is shown here again. So LSOCS, when it is edited, actually reduce the conversion of oxalate to oxalate-CoA. Thus, we can expect that ODAP content also reduced, but we could not actually see ODAP reduction in these hairy roots. There could be multiple reasons because maybe we don't have that much, the hairy roots are chimeric. So maybe still there is a residual oxalate-CoA which is good enough for ODAP synthesis or something, or maybe the hairy roots are not a good system to look at this biochemical pathway, or some other reason. So even though we could not say that it is edited and we can reduce ODAP content by genome editing this particular gene, what we can show is that genome editing is possible in Lantharis. And now we are going forward with future prospects. What are things we can do? One thing we wanted to work is we have to optimize our agrobacteria mediated transformation, that is with the attenuation. That way we can make a whole gene, whole plant transgene plants where we can edit ODAP pathway and then make plants which are the ODAP content. Also, we can so we have to optimize multiple parameters, including agrobacterium, strain, and all other methods. And also, we can even study this agrobacterium interaction with this particular probe with an idea that we can improve this transformation which can happen. Another approach we can take is a non-transformation based approach. There are multiple approaches people use for genome editing this. We can deliver genome editing constructs and all those nanoparticle mediated delivery, or virus induced gene editing method where virus mediated delivery can be tried in these Lantharis plants, or there are in-plantar transformation method where there is no tissue culture involved. We can directly cause some meristem transformation, directly applying agrobacteria into meristems and all those things. So all these methods need to be tried so that we can make this crop a better crop, all those things. Another thing we need to understand the metabolic pathway better, where we can understand how this accumulation of this ODAP in seeds, which is the most consumed part of this crop, like a seed, we can block the synthesis and accumulation in the seeds, but in the rest of the plant parts it's okay. Then we can actually make this crop a better crop. And so we need to understand more how